Usually I would be standing on this stage over here conducting the college orchestra because I'm a music professor. But tonight I'm going to talk about my moonlight activities in the field of science and how they led to a cancer research lab and an important breakthrough. Over the past 8 years, I've had the great pleasure of working with some brilliant and dedicated scientists. They were very open-minded, and we had a common dream that in the future, children would not have to suffer from cancer or from the terrible side effects of toxic drugs or radiation, because we believed there just had to be a better way. There had to be a better way, and we think we may have found it. A scientist said, "You're killing more cancer cells than as if you had used radiation." That same scientist went on, "If you had spent millions of dollars developing a new drug that killed this many cancer cells, it would be a home run." This was an astonishing thing to hear, especially for a music professor who had just completed his first experiments in a cancer lab. But we didn't use any radiation. We didn't use any drugs. So what did we do? I have here two identical tuning forks, both tuned to the note A, the note an orchestra tunes to. These forks are both made to vibrate 440 times per second. We say their frequency is 440 hertz. If I tap this fork, putting little pulses of energy into it, the second fork will also vibrate in sympathy. And if I silence this fork, we just may hear the other singing its tone. We say that I'm inducing a sympathetic resonant vibration in the second fork. It only works because both forks are tuned to the exact same frequency. Now, many of us have seen this very charming young man on the internet who shatters crystal glasses with his powerful voice. But if you watch him carefully, you'll see that first he taps the glass with his finger and listens. The glass sings its natural resonant pitch. Then he takes a deep breath and sings a loud, long note. He induces a resonant vibration in the crystal glass. The vibration grows larger and larger and larger until the glass is shattered. On the other end of the scale, we have a giant bridge made out of concrete and steel, a suspension bridge, the Tacoma Narrows Bridge. Cars and trucks and buses are going over it every day. And unfortunately, where they built this bridge, there was a steady wind blowing across it. And one day, this wind induces a small vibration in the bridge, hardly noticeable. But the frequency of the vibration matches the resonant frequency of some part of the bridge. And the vibration gets larger and larger and larger until the bridge collapses into the river below. A destructive resonant frequency. So on one end of the scale, we have a giant concrete and steel bridge destroyed by resonance. And on the other, we have a small crystal glass shattered. So maybe we could shatter something even smaller, something really small, something you would need a microscope to see. Maybe we could shatter a living microorganism. But to do that, you'd need some sort of
any peptide, anything we program the DNA to do, and it's in the living cell. Right? So in medicine, the goal in medicine now is to be able to do designer medicine and therapy. If we can design a cell to get into your body and release the right product for you you won't be losing half the drugs you take through your liver when you swallow a pill and it gets digested. These can be inserted into you through the hypospray needles, almost like Dr. McCoy on Star Trek, giving you the hyperspray. It just blasts now plasmids into your squamous cells. But Ventnor was able to do that and has the patent on the technology. But you can engineer anything. You can engineer a unique thing that would only kill one person in the world. It's how it's done. You put in a specific gene slicing, you program what you like, you put it in the cell, and it can reproduce and make as much as you like. For those of you who don't know, your DNA is usually all wrapped up in tight little coils. And so what you're doing is when they create plasmids and put them into cells, it sends a signal and tells which portion of the DNA should unwrap, unfold, and produce a product. Now, this is the future of medicine. When you look at this technology in medicine and say, this is going to be done to help people, right? We want to be able to give them medicines. We actually want to correct for genetic deficits. If a kid's born with a genetic anomaly, with the CRISPR technology, the feeling is we can create the portion of the gene they're missing and go have it spliced back in. And that may help a child, either if it's in utero development or once they're older, to have the missing substance actively produced. What would you do with this if you were in security and intelligence? Well, you can do a number of things. You could decide, if you make this gene, we know that certain people in the world who function at very high altitudes very, very well do it because they had a special mutation in their genome that we don't have because we didn't grow up in the Himalayas. But they can function at very high altitudes. Could you give this to people who are going to have to do warfighting in high altitudes and they don't require extra support? Their body makes a much more efficient use and can work under conditions of lower oxygen than the rest of us. You start letting your mind wander. Can it also produce a substance that lets you function longer underwater without oxygen? But these are run by certain mutations in genes, and with CRISPR we have the ability to actually make these and see what happens when we give them to animals, non-human or human animals, that don't have it naturally. You could have the Forrest Gump gene. You guys have been tracking. There's a gene that just makes you stronger. I would say that most of this technology is probably going to be employed by a state and not non-state actors because it's quite technical. But I say that with a caveat. When we studied Shinrikyo, if people remember, they had both uranium mines and regular laboratories where they experimented on both animals and had a whole series of laboratory experiments to develop the different kinds of gases that they wanted. Their goal was to actually mine uranium and probably come up with their own version of a nuclear weapon. But they recruited scientists, PhD level folks, and their goal was to be the rightful people running the country of Japan. But we can't assume that just because they're non-state actors, they will not make use of some technology around this. Related to this is an idea called DREADs. These are designer receptors.
And heat causes indiscriminate destruction. And we don't want that. We want targeted destruction. So we don't have to worry about heat. And now we go to the biology laboratory and we take Dr. Baer's device, and the hunt begins through a microscope for a frequency which will shatter a living microorganism. Now we have a method of controlling Dr. Baer's device by an input control frequency. So if I put in, say, 100 hertz, out will come 100 pulses per second. If I put in 200 hertz, we'll get 200 pulses. So now we're searching for the magic frequency. And we start with 100 hertz, and we look through the microscope to see if anything's happening. We watch for 5 minutes. Nothing happens. So we try 101 hertz. We look through the scope for 5 minutes, and nothing happens. So we try 102, 103, and so on. Over the course of 15 months, we try hundreds and hundreds of frequencies, if not thousands, until we find the magic combination. The answer is you have to have 2 input frequencies, one low, one high, and the higher frequency must be 11 times the lower. It's what we musicians would call the 11th harmonic. And when we add the 11th harmonic, we begin to shatter microorganisms like a crystal glass.
Of CRISPR. It's what scientists are using to engineer seedless tomatoes, monochromatic butterflies, and even extra lean piglets. CRISPR is essentially a find and replace function for DNA, and in the hands of scientists, it's a super powerful molecular tool for rewriting the code of life. So how does CRISPR work? Imagine this little toy train track is your DNA, and the genetic code that you want to swap out is this bit right here. So scientists design a short sequence of code called guide RNA that matches the bit you want to replace. Then they attach the guide RNA to a protein called Cas9. Together, they roam the cell's nucleus until they find a matching piece of DNA. Then they lock on and go to work. Cas9 unzips the DNA and slips in the guide RNA to make sure it's a match. If it is, Cas9 cuts the DNA in two. Scientists can use this to snip out a troublesome chunk of genetic code, like, say, a mutation that causes Huntington's disease. Then they can coax the cell to stitch in a new gene where the old one used to be, using the cell's natural DNA repair machinery. This is called gene editing, and it's exactly why CRISPR is such a big deal. And scientists aren't the only ones using it. Agricultural companies are designing crops that are resistant to climate change, super nutritious vegetables, and even apples that don't turn brown. Pharmaceutical firms are using it to develop new classes of antibiotics and other life-saving medicines. One day soon, doctors might even prescribe CRISPR-based therapies to treat genetic diseases like cystic fibrosis or sickle cell anemia. And Cas9 isn't the only protein that works with CRISPR. There's also Cas3, which gobbles up DNA Pac-Man style wherever it attaches. And there's one called Sherlock, which targets RNA instead of DNA and can sleuth out the presence of viruses. Designer foods, animals, drugs, these are just some of the ways CRISPR has already changed biology forever. But scientists are just beginning to explore all that this technology might one day deliver, good and bad. The question now is how we keep this world-changing technology from going off the rails.
These are the first videos taken. We showed these videos to our friends in the biology department. They said they hadn't seen anything quite like it. Seems to be a new phenomenon. These organisms are being shattered by our electronic signals. This is a harmless organism, almost friendly, a little blepharisma. And normally they're very fast swimmers, but when you approach a frequency to which they are vulnerable, they begin to slow down, then they stop, and then they begin to disintegrate within about 3 minutes. So now we know we can destroy a microorganism, and the question comes up, but can you target a specific organism with a specific frequency? So in this next video, you'll see a large organism in the center, a paramecium, undergoing disintegration, and swimming all around it, a tiny different organism, which is unharmed. If we're lucky, we'll also hear the audio as I narrate the experiment live in a noisy lab.
Perhaps you can see the sort of fireworks effect happening in the growing blister to the right of the organism. And here comes a little neighbor wondering what's going on, who's testing. And you can see blisters forming now on the lower left quadrant and upper left quadrant. The shape is now changing. And a major explosion out the top.
So now we have some evidence that we can target specific microorganisms with specific frequencies. And we make several more videos, and we film the destruction of hundreds of microorganisms. And about this time, we meet a cancer researcher, and we show them these videos. And this results in an invitation to spend 4 months in a cancer research lab trying to shatter cancer cells. This is our setup in the lab. You can see the microscope with cancer cells on it. Here's the plasma tube, and here's my little frequency control box. So first,